Video 100, Chapter 42, the immune system. All right, so as an introduction, as long-lived multicellular organisms, we need protection against pathogenic organisms. Pathogens can be a variety of different things, but essentially it means that it's something that is foreign to our body and is not really good for us, right? It can be different microbes, it can be viruses, bacteria, protozoans, fungi, parasitic animals. All of those are examples of pathogens, essentially. And although most microbes aren't pathogenic, there are some that are, and the very few that are, some of those are really good at getting past our tissues and certain barriers. And then our immune system has to kick in and fight those different pathogens that come into our body. So we're going to talk about the way that our body actually fights off these pathogens. Okay, so you do have two immune systems. You have one that you are essentially born with, an innate immune system, and this immune system we see throughout the animal kingdom. It's not unique to humans. It is what's called non-specific, which means that it's not going after specific pathogens. It's just generally protecting you. It does not have any memory, so it would not be able to protect against a certain pathogen that it's seen before, unless that pathogen is being protected normally anyways, but it's not a memory system. Versus our adaptive immune system, this one is only found in vertebrate animals. Humans do possess an adaptive immune system that is highly specific and it also has memory. So it gives us the ability to recover from an infection and then have protection when exposed to the same pathogen. So this is also where vaccines come into play. So we'll talk about both of these immune systems, starting with the innate immune system. Okay, so innate immune system, this includes physical and chemical barriers, so like your skin, normal flora that's found on your skin, the mucus and cilia in your respiratory tract, your stomach being very acidic, having a low pH, antimicrobial peptides. All of those are part of your innate immune system, those physical and chemical barriers that are, again, non-specific and do not have memory. So in the innate immune system, we do still have pathogen recognition. So any pathogen, or we'll say a microbe right now, that has different cell surface markers than our cells. So our body knows that they're not our actual cells and therefore it could be dangerous, right? Anything that's foreign and has those different cell markers is called an antigen. Okay. This lecture, just as a heads up and talking about immunity in general, can get confusing because we have antigens, antibodies, and very briefly talked about in this lecture, but something that you also know about is antibiotics. So remember, antigen is anything foreign essentially in the body. So this is kind of like pretty much the bad guy, right? And our cells recognize these pathogen-associated molecular patterns, or PAMPs, and then they do something called phagocytize. Again, these markers are not specific. They're found on a wide range of microbes. And what happens basically is that phagocytosis just means engulfing. So it's basically like little Pac-Man. So they see something foreign, like a little bacterium here. You'll see the receptors basically recognize it and tell a phagosome or to phagocytize, which means that it's going to engulf it and then go through a process of essentially digesting that bacterium or antigen, whatever it is, and then putting out the debris from it, okay, which is then going into exocytosis. So we'll see later that cells of the adaptive immune response can recognize one bacterial species from another, and they have different responses to that. But within the innate immune system, they just have general phagocytes that are going after these things. So different cells that are involved in the immune system. I know this might be hard to read on the slide, but it is obviously in your book. And the cells that we're looking at here are white blood cells and leukocytes, so WBCs and leukocytes. You can see here we have mast cells. Macrophages are one of the main ones that we really talk about within the immune system, and it's the cells that are phagocytic, and they're essentially consuming foreign pathogens as well as cancer cells, and they're also stimulating response of other immune cells. Okay. Another kind of cool one, I think it's got a cool name, is a natural killer cell, or you'll hear them called NK cells sometimes. These guys can actually kill your own cells if they're infected. So this is something that we want if we have a tumor or a virus-infected cells, maybe cancer. If it's infected, your natural killer cells will actually still go after your own cells, and they'll do it by doing something called triggering something called apoptosis. Apoptosis is basically cell death. Usually it's like cell suicide is kind of what we refer to it. So natural killer cells will trigger apoptosis. We have dendritic cells, monocytes, neutrophils are another big one. I mean, all of these are well involved, but I think some of the main ones that you'll hear about are what I'm highlighting here. And neutrophils is definitely one of those. It's one of the first responders at the site of an infection or a trauma, and it's very abundant, right? Neutrophils are we have the largest amount of neutrophils in our system. Well, I should say they're the most abundant white blood cell. And you'll see these when they go and attack something, like say a site of infection. If you cut yourself on something, it's releasing toxins that kill or inhibit bacteria and fungi, and it also recruits other immune cells to that site of infection. And these you see when they do their job, they go through and they actually die off, and we see them as pus, basically. So neutrophils are one of our more abundant parts of the innate immune system as well. We also have basophils and eosinophils. I'll let you folks read about those, but this is part of the innate immune system. So these are things that are happening again as a physical or chemical barrier that do not have memory or any specificity to them. They are usually involved in an inflammatory response. So with inflammation, like if you cut yourself or you hit yourself pretty hard on something, with that comes pain, usually too near the site of infection, or if there's a cut, you'll have some heat and your body temperature is going to raise in an attempt to slow the growth of the pathogenic bacteria that may be entering from that wound. You're also going to see redness and usually swelling, and the redness and swelling are due to increased vascular permeability to allow the white blood cells to leave capillaries and enter the tissues. So what happens is you have some blood vessels that are essentially vasodilating, which means that they're becoming larger, and they're increasing that permeability, allowing more white blood cells to come in. So this is where we get histamine responses from as well. So even though this doesn't always look good, when you see a lot of redness or swelling around a cut that you might have, it actually means that your innate immune system is working well. So it is technically a good thing. It's something that we want to see. And you'll feel sometimes around like your wound, it might feel like kind of hard or obviously painful to the touch. And again, that's going through this whole process and calling in some of these cells to come over and kill foreign antigens that might be coming in. Again, just a quick recap on your natural killer cells. These are the ones that can kill the virus-infected or altered self cells or tumor cells. So they're coming in and they're basically putting a little puncture into your cell, and that is what is leading to the apoptosis or the cell death. A little bit more background on this. These natural killer cells, you'll see here this little green thing called MHC1. MHC1 stands for Major Histocompatibility Complex 1 molecules, and they are on the surface of infected cells. And basically what they are, they're proteins, and their whole reason that they're here is to display fragments of proteins from infectious agents within the cells, and they're showing that off to T cells. So T cells are just lymphocytes that mature in the thymus gland. You'll also hear about B cells, which are lymphocytes that mature in the bone marrow. So they're appropriately named T for thymus, B for bone marrow. That's where they mature. But they're cells of the adaptive immune system that we're getting to in just a second. But this MHC1 molecule, and we also have these things called MHC2 molecules, but MHC1 molecules here are basically telling other parts of the immune system, like, hey, look, I'm not what we want in our body. And so that's what they are. And then we also have MHC2 molecules that are that contain antigens, and they're on lymphocytes as well. And those interact with helper T cells to trigger immune responses within the adaptive immune system that we'll talk about here in a second. All right, so getting into the adaptive immune response, I did post a video that might be helpful. Again, it's Amoeba Sisters. There's lots of other helpful ones. Crash Course is also usually helpful, but Amoeba Sisters kind of takes some of the main highlights and makes it really quick. So I like the Amoeba Sisters videos because they go through a lot of information a little bit quicker and kind of condense it. But feel free to look at that and kind of understand the immune system a little bit better. This is a very complicated system that you could have a whole class on, just like every one of our lectures, really. So we're just giving you a little bit of a taste of it. But now we're getting into the adaptive immune response. And again, a few different kinds of definitions that you'll need. So we have two different kinds of cells in here, our lymphocytes, B cells, again, bone marrow, T cells, thymus. And with the adaptive immune response, remember, this is specific and it has memory. So because of that, it can take days or even weeks to become what's called upregulated, which means essentially it can take days or weeks to start working. Whereas our innate response that we just finished talking about, that one works immediately, like a physical or chemical barrier, right? That is working since birth. And although we can have adaptive immune responses, going into babies as well from the mother going through the placenta, antibodies can be passed down. The babies can actually lose that. So that's why kids are constantly getting sick, is because they're building up these immune systems and getting all of their adaptive immune response going so that they can have that memory when they start seeing these bacteria or viruses, whatever the antigen is, again. So adaptive immune response, you'll see here in a second, is divided by these cells, essentially. So we have a cell-mediated response, which is carried out by the T cells. And then we have what's called as a humoral immune response, and that one is activated by B cells and antibodies. Okay, so characteristics of our adaptive immune system. We have highly specific antibodies, again, made by B cells and T cell receptors, or TCRs, that are made by T cells. And this innate immune system recognizes those PAMPs that we talked about earlier that are found on a variety of microbes. But then we have these B cells and T cells that can come in and can distinguish between different motifs or epitopes that are found on an individual microbe. So we're just getting to a higher level of specificity here where we can really differentiate between different kinds of microbes, and we have the memory to attack them and to react a little bit quicker than normal. So again, adaptive immune response takes some time when you're first introduced to an antigen, but once you've been introduced to that antigen, then your secondary response to it is going to be much quicker. So this is why we have things like vaccines, or if you have chicken pox one time, you're usually set for the rest of your life because now your body recognizes that and can fight it off if it encounters it again. So this is all part of your adaptive immune response. Okay, so first talking about our humoral response. So this is where our B cells come into place. Again, they're from the bone marrow, and they are making antibodies, or ABS is how you'll see it abbreviated from time to time. Remember, antigens are our bad foreign guys. Antibodies are the guys that are actually going to help us. They're proteins that are encoded for in our DNA. So our DNA is making very specific antibodies. And each B cell is producing antibodies with a specific variable region that recognizes a specific epitope. Or essentially just think of it as recognizing a specific antigen, really. So these can be secreted or membrane bound on the B cell. And essentially what's happening is our antibody here is going to recognize and bind to very specific antigens. Again, remember that specificity and that memory. So each B cell secretes antibodies with that specific variable region that's going to recognize a specific epitope. And we have different kinds of antibodies, and you might have heard of these before, like IgA, IgD, IgE, IgG, IgM. I think some of the ones that you hear about most are like IgE. This protects against parasitic worms. It's also responsible for allergic reactions. IgG as well might be one that you hear quite often, and this one's secreted by plasma cells in the blood. These antibodies are the ones that are able to cross the placenta into the fetus. Not all of them can. And you can go through the others too. I think those are two of the ones that we probably hear about the most. But we do have other antibodies in here as well, like IgA, which is found in our mucus, saliva, tears, and breast milk, that protects against pathogens. Our IgD will activate basophils and mast cells. Our IgM can attach to the surface of a B cell and be secreted into the blood. This is our early stages of immunity. So these work at different stages and with different antigens as well. Okay, so how do antibodies actually function? What's going on here? So there's a few different ways antibodies can be working. One is through something called neutralization, and this is where they prevent a virus or toxic protein from binding their target. And this is literally like they're physically blocking the binding sites on viruses or bacterial toxins, whatever it might be. So you can see the antibody here. And again, it's just literally blocking this virus from really being able to go to another target cell. So neutralization, physically blocking, it's like you're boxing them out in basketball type of thing. Opsonization, this one is a little bit different. This is like free-floating antibodies that are kind of roaming around, and they're consumed by a macrophage or a neutrophil. So you can see your pathogen here and our antibodies that were free-floating kind of attach here, and then they call in the macrophage. So a macrophage is basically just a big cell that goes through phagocytosis, essentially. So it's going to surround and kill the microorganisms, and then it's going to go through that little digestive process and remove the dead cells and stimulate other immune cells to come in as well. And then up last, we have complement activation. So this is where our antibodies are attaching to the surface of a pathogen cell and activating what's known as the complement system. And the complement system is essentially, it's allowing other complement proteins to bind to it. So the combination of antibodies and complement enhances the opsonization that's occurring. So when a macrophage comes over and kind of engulfs it and goes through that whole process, so it can further promote and rapidly clear any pathogens that are coming that way. So again, our complement process, it can kind of essentially work to make a hole in the pathogen's membrane. So you'll see the invading pathogen here, the antigen that is on that pathogen. So an antigen, another way to kind of describe it, it is a foreign material that's coming into your body, but a lot of times it's essentially like a flag or a marker that is on an invading pathogen. So you'll see here our little antigen, and that is connecting up with our antibody that is meant for it. And then what's going to happen is another protein, in this case C1, is going to bind to that antigen antibody complex on the invading pathogen, and it's going to cause complementary components, which are C2 and C4 here. Those are going to split in two, and then they're going to go through this whole thing where they convert and split into other molecules as well. And so you have all of them combining and forming this whole complex that's going to make a hole in the pathogen's plasma membrane, which causes water to rush in and causes the pathogen to lyse or essentially explode. So that's how the complement system works. It's essentially just bringing in other complements or other proteins to help knock out this invading pathogen. Okay, so we talked a little bit about these MHC, again, MHC1 and MHC2. They're antigen presenting molecules. MHC1 is found on all of our nucleated cells, which is pretty much all of our cells minus our red blood cells. And then MHC class 2, these are found on antigen presenting cells, or APCs. These are things like our B cells, our dendritic cells, macrophages. And to go over them a little bit more, like dendritic cells, these are immune cells that can process antigen material. They're present in the skin. They're also in the nose, your lungs, stomach, intestines. And sometimes they present on the surface of other cells too. Just depends on what APC they're attaching to, basically. Macrophages also function as APCs. And before activation and differentiation, B cells do as well. So after phagocytosis by APCs, the phagocytic vesicle will fuse with an intracellular lysosome. Lysosome is just, again, digesting any of the cell contents that it's going through. So it's breaking down the pathogen or, whatever is being phagocytosed and forming something different. Okay, so then you can break down these components and then they're loaded into either MHC1 or MHC2 molecules and transported to the surface to be able to throw out their little antigen flag and present it. Okay. Okay, so now we get into our cell-mediated response. So we went over the humoral response, which is essentially our B cells, our lymphocytes, or our B cells are a lymphocyte, but so is our T cell. And the humoral response involved our antibodies. Now we're getting into the cell-mediated response, and this is with our T cells. So these, again, are coming from the thymus. This is a little bit of a different response. So these guys are producing T cells receptors, or TCRs, that are membrane bound. Again, these are encoded in our DNA, and they recognize specific antigens presented in MHC molecules. Okay, so T cells there's three different types of populations. There's helper T cells, cytotoxic T cells, and regulatory T cells. But essentially what T cells are doing is they're causing inflammation, they're activating macrophages, they're getting other T cells to get kind of fired up, they're regulating, which is our regulator cells, and they're regulating a lot of the immune response, essentially. Okay. So we'll get into how these antigen presenting cells, these APCs, process antigens to be presented on the MHC to a T cell. So you'll see here a bacterium engulfed by a macrophage is encased within a vacuole. Lysosomes fuse with the vacuole and digest the bacterium. And then what comes out of that after digesting all of this stuff in here is we get some of these antigens that can be presented on these MHC2 molecules. So we are talking mostly about MHC1 class before this. Now we're going to MHC2, and the antigens, again, are on this MHC2 cell surface. So after we go through that process, then we get into the MHC2 molecules are actually going to call in the helper T cells. Okay, so you can see here when a CD4 plus helper T cell, you can just call them a helper T cell, binds to that MHC2 antigen complex, both the antigen presenting cell, the APC, and the T cell release what are called cytokines. Okay, cytokines are essentially different kinds of substances that can interfere with or have an effect on parts of the immune system or affect other cells. So these guys will release these cytokines, and in response to the cytokines, we have T cells that actually clone themselves. So now they're starting to clone themselves. And the clone T cells are going to produce different cytokines that then activate the B cells that we just talked about in our humoral response, as well as CD8 plus T cells. And when they clone, they're pretty much cloning and getting all different kinds of T cells. So they're getting more helper cells that can activate cells that kill. They're getting more cytotoxic T cells that are doing the killing, as well as some of the regulatory T cells that are helping regulate this response. So you can see here cytotoxic T cells, these are the ones that actually kill the infected self cells or altered tumor self cells. So these are the ones that can go through and kill, whether it's foreign or your self cell. And for instance, if a cell is infected with a virus, it's going to present that virus on its MHC class 1. And then your cytotoxic T cell or your T sub C cell will then force that self cell to go through apoptosis. Again, kind of a programmed self or suicidal cell death. And that's how the cytotoxic T cells are working. So how does the variable region of an antibody secreted by one B cell differ from another B cell? How does the variable region of a T cell receptor of one T cell differ from another T cell? So in order for these to be different and to recognize different antigens within the immune system or within your body, I guess, we have to go through a process called gene rearrangement. So while these B and T cells are developing, they're developing in the region of the DNA that codes for variable regions, and it rearranges itself so that each B and T cell have a unique variable region, which means that we're going to get really high diversity and we'll essentially be able to recognize many different kinds of antigens and have lots of different kinds of cells, which is great because then we can help further fight off these pathogens that are coming into our body. Again, this process is really complicated. It's not something that we'll go into in very much depth in this class, and this lecture is even a little bit less depth than your reading goes into. So in your reading, that's going to help to kind of understand some of this, and you might see some things that we didn't talk as much about. I wouldn't worry about some of the extra details. I want you to kind of know the general gist of how your immune system is working. All right, so we also have something called a lymphatic system. This is another part of the immune system, and the lymph is a watery fluid that's traveling from your lymph vessels to be dumped back into circulation. Most of the cells in your blood vessels we know are erythrocytes or are red blood cells. And then we also have something called leukocytes, right, as well as white blood cells, and they can migrate into tissues and fluid, and they can go with nutrients, they can go with everything, right, as well as the lymph. So along the way, all this lymph that's being carried around is traveling through something called lymph nodes. And that is where we have a congregation of BT and other white blood cells. So if something is foreign and it made it past the skin or mucosal membranes into your tissue, so it's gone past your innate immune system, basically, it will hopefully be detected in the lymph nodes. And that's where we get more clones of T and B cells that are specific for the antigen being released into circulation to find more of that same antigen. So you'll hear sometimes like, oh, I have some swollen lymph nodes. Again, that's usually, it's not a good sign in terms of you probably do have a hopefully minor infection, but it's a good sign in that it means your immune system is working through the lymphatic system, and that hopefully what's happening is that your B and T cells are cloning each other, or cloning one another. Essentially, your B cells are cloning, your T cells are cloning, and they're sending out more helper cells, more cytotoxic T cells, and that's going through your whole system so that your whole body can fight off the pathogen that's coming in, whether that's like a flu virus or some bacterium. That's what's happening in your lymphatic system. Okay, so we have a primary and a secondary response with our adaptive immune system, and that's why we say that it takes so long to build it up, right? You'll see this graph over here, concentration of the antibody over time. So we don't have an antibody to begin with. Let's say we're exposed to the flu this year, right? And when we first are exposed to it, we've never seen this before. This is something new. So our body's got to figure out, okay, how am I going to respond? Am I going to activate the humoral response? Am I going to activate our cell mediated? Is my innate response enough? No, definitely not. So it's going to take some time to build that up. And then you have this primary immune response that has low affinity antibodies. Okay. So what's happening is it took a lot of time, but now we're going to build up these antibodies, and then we're going to kill off the infection, and then you'll see them go back down, but they don't go back down to the initial, right? They're still elevated some, so that when we do hit a secondary immune response, our memory cells are going to respond a lot faster. Okay. So again, you'll see with this primary response, this can take days to weeks for your B and T cells to upregulate and mount that full response. And then after recovery, we're going here. And with the secondary exposure, then we're going to respond a lot faster, and we're going to build up a lot more antibodies really quickly. So you can see this big spike in building up the antibodies once let's say we see that flu again, or let's say that we got our flu shot and we have our primary immune response there, so that when we're actually exposed to the flu again, we have that uptick in upregulation of B and T cells. And that is specific to that virus. So now what happens is you're either going to have less symptoms, or you're not actually going to get sick, or you might recover faster if you do. So that's why we have this primary and then secondary. This is exactly how vaccines work. That's how it works if you get certain other pathogens, like say you got the flu, whether you got a vaccine or say you're exposed to it from someone at work or your kid, and maybe it makes you sick, or maybe you can kind of skirt it a little bit, and then when you find it again, then you're going to have a much better response. So vaccination, again, is our primary immune response. That's what it's initiating. It's giving the body time to mount a primary response against a pathogen, hopefully before you'll see it again, so that you can make some of those memory B and T cells against that specific pathogen. And then when you're exposed later, hopefully it's going to prevent sickness, or in a lot of cases, it can't necessarily prevent sickness, but it can help you recover faster or have much more mild symptoms. Again, some of the different antibodies that we have: IgA, IgD, IgE, IgG, IgM. All of these are kind of involved in different things throughout the body, and I'll let you guys read a little bit more about that and see more about that as well. But with our antibodies, we have something called affinity versus avidity. So affinity just means like the strength of one single antibody to antigen interaction. So for instance, IgG, antigen binding site, typically has a really high affinity for its target, meaning that it's really good at getting to its target. Whereas avidity refers to the strength of all of those interactions combined. So something like IgM typically has a really low affinity. It's not as good as IgG at binding to the antigens, but there's 10 of them. So the avidity is really high, and it's therefore also really effective. We also have cross-reactivity with some antibodies, which means that it can react with two different epitopes. Again, epitopes are basically like small components of antigens that are presented by those antigen-presenting cells. So this is something we look for in the antibodies, whether they have high affinity or high avidity, as well as cross-reactivity. Those are all kind of the ideal in your antibodies, right? You want them to be able to bind really well, or have lots of different sites, and or be able to work with different antigens. So things that can go wrong. Obviously, if you have chronic inflammation, that can start to cause damage to your tissues. Allergies, if you're allergic to pets or certain foods, that means your immune system's overreacting to that antigen. And then there's certain antibodies that are involved in each one of these. And then we also have autoimmune disorders, right? And this is where you start to recognize auto, meaning self, basically. You start to recognize self antigens as being foreign. So things that are in your body regularly, now our body thinks that it shouldn't be there and it's going to attack them. And that's resulting in tissue and organ damage. Some autoimmune disorders that you might have heard of before: lupus, multiple sclerosis, Graves' disease, even rheumatoid arthritis. All of those are autoimmune diseases that essentially have a problem recognizing self from foreign. There's also things called having to do with immunodeficiency, and there's a primary immunodeficiency, which is genetic, or it could be developmental. And it basically means that you're lacking some component of the immune system. So some diseases that you might have heard of are immunodeficiencies like SCID, inability to make a certain antibody class, whether you can't make IgA or IgM, and also a lack of a thymus, which could be a problem because what's made in the thymus are T cells, right? And then sometimes we can also acquire an immune deficiency later in life, which would be like AIDS, right? AIDS is a result of a HIV infection. AIDS AIDS essentially attacks our helper Ts, and our helper Ts are involved in, finding more T cells to essentially attack and kill foreign invaders. So it means that now we're immunodeficient and we don't have that ability to find and kill more more pathogens that are coming in, or different antigens on different pathogens, and so we're not going to be able to fight anything off. And then allergies, the way allergies work, you can kind of see here, if we have like a pollen, let's say, that we're allergic to. And what happens is that pollen, or whatever you might be allergic to, some potential allergen, they're then going to synthesize antibodies of the IgE class via your typical process of antigen presenting cells presenting processed antigen to your helper T cells. That's going to stimulate your B cells to produce IgE. Okay, so after that, your IgE antibody is produced, and it's going to attach to things called mast cells. These mast cells then are going to release basically modified amino acids, histamines, and serotonin as well. And then those chemical mediators recruit what's known as eosinophils. And that's what's actually targeting or coming up with your allergic responses. Okay, so you can see that the effects of an allergic reaction can go anywhere from mild symptoms like sneezing or itchy, watery eyes to life-threatening and maybe anaphylactic. Okay, so the way that we treat these obviously is different depending on the severity of the allergy. If it's super severe and you're anaphylactic, that's when you need epinephrine. You need epinephrine pen, basically, so that your allergy is not fatal. But this is how allergies are working. So if you have allergies to different things, this is essentially the process that your body is going through. And that's going to be it for immune response. Again, watch that video. It's probably going to help. I know that the difference between antigen, antibody, and even getting into antibiotics, which we don't cover here, but we know that antibiotics are helping to target bacterial infections, and they do that by doing kind of the same thing that some of these immune system cells do, is essentially poking a hole in the cell, or what antibiotics are really doing is they're going after the cell wall of bacteria and causing those cells to burst or lyse. So there's a lot of very similar terminology in this. Don't get caught up in it. Make sure you take good notes and that you watch those helper videos as well. And let me know if you have any questions.